So over to you, Dayo.
Okay, I hope everyone can hear me. So I'd like to first start by saying thank you for making the time. I'm really excited to talk about intra-African bird migration, focusing on the woodland kingfisher. This is part of a postdoc that was a postdoc fellowship that was done at the Fitzpatrick Institute of African Ornithology at the University of Cape Town. And my PIs were Professor Peter Ryan and Phoebe Barnard. And I also collaborated strongly with Desiree Dalton, who at the time was at the National Zoological Gardens in Pretoria. But again, a lot of this work is brought together more than these four individuals. So I thought that it would be nice to start with some woodland photos, woodland kingfisher photos that I've sort of gathered over the course of the project. They are really, really beautiful birds. You can really see them, but you can also hear them. And so this is one kingfisher displaying. All these photos are mine, taken in the field. Yeah, I find them. I think they're really handsome birds. Some will say beautiful, and they're really great study species. And in this particular photo, you can see this individual is clearly ringed with a metal ring, with an aluminum ring. But we also used color rings for some individuals during the course of the study. But of course, you need to get the angle right to be able to see the tarsus of the bird. For the keen-eyed birders among you, you would probably realize that this kingfisher is not a South African one because it doesn't have that eye stripe, the black eye stripe behind the eye. I will come to that. That is of significance, and we'll talk about that in a short second. So I'm just going to let this video run while I just give a broader introduction to the project itself. So I started the postdoc in 2015. We were able to get some startup funding from IFS, the International Foundation for Science, and there was also funding coming from UCT itself, from the Fitzpatrick, to get this work started. And very little. And we know a lot about inter-African migrants. So the Nearctic birds moving between North and South America, as well as the Palearctic birds moving between Europe, Asia, and Africa. But within continents, and not just the African continent now, we don't quite know so much. And there are a lot of data gaps sort of there. So in my former life, I had worked with BirdLife to work on, to develop the Africa-Eurasia Migratory Land Birds Action Plan. And this focused a lot on migration as defined by CMS, the Convention on Migratory Species. So moving sort of with that, I would like to I just want to make sure that is not visible. Good. So when we talk about migration now, just so that we're all clear about this, migration is movement. It does involve movement. Some would say, is it between space or is it between time? It generally, it is across space and time. So the animal has to move from point A to point B. The movement also has to be predictable. So it's not just the animal being a vagrant. So it needs to be seasonal or diurnal. So you can have daily migration in some cases. Then the movement also needs to be cyclic, which means that animal needs to move from point A to point B or C and back to point A for it to be considered migration. Otherwise, you're just going, just wandering around. Again, that would be vagarism. In some cases, that cyclic movement is made by an individual, but in some cases, it is made across generations. And one of the classic examples is that of the monarch butterfly that migrates between South and North America across several generations and back again. So it's just worth mentioning that. And the last criteria or point for migration is that it needs to be benefit-based. So you're either avoiding predators or you're gaining resources. You could also be avoiding parasites in certain cases. So you need to be able to sort of identify these 4 elements to be able to say that, yes, this animal is actually migrating. And then just to still talking broadly about migration, when we talk about birds, we talk about the flyways, which I like to call the highway for birds, and there are a lot of many of the flyways have been acknowledged. This is imagery available from BirdLife, and it gives the names of the major flyways. Now, you'll notice that a lot of them are across continents. I want to talk about the movement of birds within continents now. So as said, one of the study species that during the postdoc was the woodland kingfisher. And this is the global range. It's found just in Africa. There are 3 subspecies. So Halcyon senegalensis, Halcyon senegalensis physiopelius, and Halcyon senegalensis cyanoluca. What makes it nice and interesting is that they have sort of slightly different ranges. So this is the range of the senegalensis, that's senegalensis subspecies, where the green areas is where it is found to be resident all year round, and the yellow is where it migrates into seasonally and comes sort of south, at least in the case of the senegalensis. You have the physiopelius, which is largely sedentary from what is available in literature. It is not one of the migrants in the woodland when we talk about woodland kingfishers. And then we have the cyanoluca, which is an interesting one because you've got this band where it is resident, and then to the north of it, it migrates. So it migrates south into South Africa to breed, and then it goes north. It makes a trans-equatorial migration, and we are yet to. It's supposedly it doesn't breed when it's up there, but I'm going to say that the jury's still out on that, and I'll explain why in a short second. But when we put it all together, we get this really interesting mishmash. And most people just see kingfishers, woodland kingfisher, and they're like, oh, I saw woodland kingfisher. And you can find them, anywhere across, almost anywhere across sub-Saharan Africa. But in this study, we started looking closer at them and trying to tease themes apart. Now, we established, we took a broad sort of spatial approach to this project. So even though I was based in South Africa, we established field sites in Uganda, in Nigeria, and in Ghana. And we tried to access these field sites during the breeding season at the site. So at the peak of the breeding season. So between May and June, I was in West Africa. July, August, I was in East Africa in Uganda, and then November to January, I was in the Limpopo province area of South Africa. We did have a lot of help, and I do want to acknowledge the support of a lot of the organizations that helped us when we were on site, the organizations that we collaborated with, that we worked with. Yeah, so in terms of the work itself now, coming into the sort of more technical side of things, the first thing, of course, is trapping the bird. And we used mist nets. That was our primary method for trapping birds. And I take it that many of the people that I'm talking to are very familiar with mist nets. We had to get very creative with our mist nets. So this is what we call a double-decker T-shaped net array. I think we ended up using, wait, I have to double check. 1, 2, 3, 4, 5, 6 mist nets in this array, and it really did help with increasing our capture rate of not just the kingfishers, but many of the other species that we worked with. And I'm working on a method paper for this this double-decker net array where we're using high nets, but we're not using a pulley system. And people who have worked with mist nets, particularly in areas where you've got thorns, you will very much understand how interesting it can be, or how fraught the process can be with trying to make sure that your nets don't get blown onto the thorn bush. I just heard a sound. Is there a comment? Okay, good. I'll just continue. Okay, so I thought I would just share with you, I like to give people that experience of being in the field. So I have a video here of our efforts to trap a kingfisher.
Good catch.
So in that video, when our trapping is very sort of focused. We get to a site, we try to identify the territory of the bird, because we're usually doing this during the breeding season, and then we put up the nets in what we think is the best place, and we usually use playbacks as well to bring them a little bit closer to the mist net. Here is another one. This was in Ghana. The first one was in South Africa.
That's it.
There's a lot of excitement when we get the birds in the net. Just because many of them just overfly the net or go around it. So focal trapping can be quite interesting. We expanded our methods, and we also started using spring traps as well. In this particular case, we were actually fortunate enough to catch both the male and the female, the pair, in this territory. So yes, I had to take a photo of the birds sort of side by side. And with the spring traps, we bait the trap with a superworm just over here, as you can see. And yeah, so with all that, with those two methods, looking just at the woodland kingfisher, we were able to get 10 birds in Ghana. We were able to trap 12 in Uganda. So 2 sites in Ghana, so in Accra and in Cape Coast. In Uganda, we trapped all 12 in Entebbe. And in South Africa, across 3 sites, we were able to get about 58 birds, of which 17 were retraps. Now, one of the reasons why trapping in South Africa was much more successful was because I could go into the field with more equipment. So normally what would happen is in November, I would gear up at the university. I'll take a bucky and just load it full of gear and then head out to Limpopo. We were also able to put up nest boxes in some of the at one of the sites in Limpopo, and that also did help with knowing where the birds are and sort of being able to put the mist nets in or spring traps in the right place. All birds that were trapped were ringed, and in many cases, we did also use color rings so that we could identify the birds when they were perched. And all the data from the project was uploaded to the SAFRing database. Once we had the bird in the hand, we did take all sorts of all the measurements, and even the retraps. We did take all of these measurements from the birds. So now, in terms of our research questions, we tried to focus on four main areas. We were trying to look at variation in woodland kingfisher across the range. We looked at phylogeography, so basically gene flow across the continent, then phenology, so timing of movement, and then we looked at migratory connectivity, how sites are connected for the birds that are actually moving. What sites are they going to, and yeah, are they coming right back? Are they connecting particular sites? So with the variation, one of the areas that I mentioned earlier, that the South African Cyanoleuca has this eye stripe, which the Fusciopelius and the senegalensis do not have. So this is a kingfisher from Ghana, kingfisher from Uganda, and this is a juvenile one from Uganda. They don't have this eye stripe, but the South African Cyanoleuca, I call it the South African breeding Cyanoleuca does have this. So without even having the bird in the hand or even looking at the genetics, you can spot this bird quite easily. We haven't done it yet, but there is a project also coming out of at the Fitzpatrick. It's called Morphometrics, where they make use of a lot of online imagery and with metadata, and sort of use that to sort of give geolocation of where these birds might be. So sort of using social media to help with movement of birds. So we sort of think that the program, the algorithm, should be able to identify the eye stripe, and the woodland kingfisher might be an ideal species, just to tease apart the Cyanoleuca from the other, the senegalensis and the Fusciopelius. And that might also give us an indication of the movement or the range of the Cyanoleuca. But this is a project that we would like to sort of do, work with the Morphic team, but that hasn't happened yet. But yes, we would like to see if we could play with this. The other variation is vocalization. And I'm just going to shut up for a quick second and let you listen to the call and look at the call of the woodland kingfisher. So this is the call of one individual, and this is the characteristic call of the woodland kingfisher. You have that first, and then you have the trills that follow it. And in the case, looking at the spectrogram, you notice there were about three layers when you look at it sort of vertically. The other two are what you call harmonics, but I won't sort of get into that. So as part of a preliminary assessment, we took some of the early recordings that were made at the start of this project in Ghana, South Africa, and Uganda. And we looked at the duration of the call. That is from the first strip to the end of the last trill. how long in time, how many seconds that is. We looked at the trill duration. So that is that each one of them. We painstakingly measured how long each trill was. And then we also looked at the number of trills. And then we compared this across Ghana, recordings from Ghana, recordings from South Africa, and recordings from Uganda. Now, the statistics on this hasn't been done. We are, I will say up front that it would be awesome to maybe, if a master's student is interested in this kind of a project, all the readings are, all the recordings are available right now, and we would love to be able to sort of settle down and look into this with the entire data set, and then statistically look at differences. But the preliminary assessment shows that, particularly between the Southern African recordings and the Ugandan, there is room for, like, there's a big difference where the birds in Uganda were calling for longer, and they had more trills, and each trill was also slightly longer. Now, vocalization for a lot of birds, not all, but for a lot of birds, can also be used as a mark of fitness. So it's sort of used in competition. So we're quite interested in what this might mean across subspecies. Now, you'll notice that in Ghana, the variation is quite wide. Right at present, my leading thought is, if you remember the Physiopelius, the range does overlap quite strongly with the senegalensis in West Africa. My thinking is that maybe we were recording, maybe we had some Physiopelius recordings and we had some senegalensis recordings as well. Now, with the vocalization, we didn't always trap the bird. So whenever we just saw a woodland kingfisher singing, we would just record take as much recording as we can. The other thing with playbacks that we tried, with the recordings that we tried to sort of do to control the purity of the data, is that we would do the recordings before we start trying to trap birds in a particular area, because we didn't want the playbacks to influence what the birds were singing or what they normally sound like. So normally our protocol is, when we get to a particular site, first couple of days, we go out and we try to do as much recording as we can to eliminate that latency effect of using other playbacks, because I've seen birds try to match the playback that we are using, which generally means that the bird is changing the way it would naturally call. So that is, in a nutshell, just talking a little bit about variation. Now, jumping into phylogeography. So we took blood samples from all the birds that we trapped and that we sampled, and for those interested, the blood was taken from the brachial vein of the bird, and the blood was stored, and we worked with the National Zoological Gardens, the biobank, and all the data, all the tissue samples were stored there. So what we wanted to look at was mitochondrial DNA and nuclear DNA. Mitochondrial DNA for many mammalian species. I'm not a geneticist, but the little that I've understood is that it's passed down along the maternal line, and the mitochondrial DNA speaks to more ancestral lineages, whereas the nuclear DNA is more it's more contemporary, and it speaks to variation in a shorter sort of timeline. Now, I say shorter, but we're still talking thousands of sort of years. So with the mitochondrial DNA, what we expected was that the looking across the subspecies now, the Cyanoluca, the senegalensis, what we were expecting was that, oh, ancestrally they were all one, and then they were split. So I was expecting that the mitochondrial DNA, this is showing preliminary results from looking at South Africa and Ghana. So the green ones are samples from Ghana, the yellow are samples from South Africa. I was expecting that using the mitochondrial markers, they would be closer together, and using the nuclear, they would be, split. But what we actually found, which is interesting, was the opposite. So using, this is the first mitochondrial marker. It showed that the Ghana and the South African populations are very distinct in terms of the mitochondrial DNA. We used a second marker, and it showed exactly the same thing or similar results. And then we got to the nuclear, and boom, they were kind of, in terms of contemporary time, they were actually kind of closer together. So this got really interesting for us, and we looked at it in greater detail. I say we, working with geneticists. So we have just had a paper accepted now for publication in Ostrich. It's titled Genetic and Morphological Variation of Halcyon senegalensis, Revealing Cryptic Mitochondrial Lineages and Patterns of Mitochondrial Nuclear Discordance. And one of the highlights of this paper is that we can actually date when they split, when the senegalensis and the Cyanoluca actually split. So that is an interesting paper that I would say for those interested, just keep an eye out for it. It will be published in Ostrich soon enough. But working with the National Zoological Gardens, there was an earlier paper that was looking at avian hemoparasites. And this was just a broad one, looking at what hemoparasites we found. So for those that might be interested as well, this paper is available, and it came from this study. So moving on to phenology. Using data from the SABAP2 database, we looked at the reporting rates for woodland kingfishers in Southern Africa, not just in South Africa, but in Southern Africa. And the blue line shows the expected. So looking at, so from July all the way through to June, the blue line showed the expected, so these are the reporting rates, where we had a big spike in reporting rates around November, December, which is generally, so from sort of October, November is when the birds begin to arrive. And then from around April, May is when they leave Southern Africa. So what we noticed was that, and I'm taking these 3 years, 2014, 2015, and 2016. So the red is the actual. In 2014 and in the years before that, we found this trend where the birds were sort of arriving either on the expected or a little earlier than expected, and they were leaving later or around the date that was expected. In 2015, it was sort of roughly matched. In 2016, the birds arrived later than expected and left earlier than expected. Now, this is really interesting when we start talking about climate and land use and what are the drivers for migration within the African continent. My understanding from talking to many from the South Africans that I've interacted with was that 2016 was a drought year. And it would be interesting to explore this data set a little bit more, see if we have any other years when this happened, where the woodland kingfishers stayed in South Africa for a short amount of a short period. But what we would like to do, we haven't done it yet. What we would like to do, and again, with this, I'm also casting out a wide net. If we have any students that are interested in this kind of research project or anyone that would want to collaborate, what we're hoping to do is take the SABAP2 data and merge it with what's his name, remote sensed data like the NDVI data, vegetation mapping data, and look at land changes across southern Africa, but also looking at where the birds might be at a particular time. Because the question is, how do the birds know? So is the drought just localized in South Africa, or are the birds aware of it as they're migrating down? So we have not yet looked into this, but we think that this would be interesting, and it would look at what it would help us identify factors that are driving the arrival and departure dates of migrants within Africa. So if anyone has any thoughts, I would definitely love to hear them at the end of the presentation. But that is what we were sort of looking at in terms of phenology. Now, just lastly, connectivity. So just to sort of bring it home for just to explain again. So in terms of migratory connectivity, there are a couple of examples. You can have weak connectivity where individuals from a particular site, so say we pick this site in South Africa, they go to different sites. So they sort of disperse, but they all come back to this one site. But you could also have strong connectivity where all the individuals from one site, this is the example marked in orange, where individuals from one site all go to another site, and then from that site B, all come back to site A. So in that case, you would say there is strong connectivity or strong connection between site A and B. And when they're going to a bunch of different sites, then you would say there is the weak connectivity. So we wanted to look at our woodland kingfishers connecting sites across Africa strongly or not quite as strong, not quite as strong. So to do this, one of the methods we employed was using geolocators. Now, for those that might know a little bit about telemetry or biologging, there is this arbitrary number that is being put out there, 3 to 5% of an animal's body weight. So if you're going to put some sort of a device on an animal, it is recommended that it not weigh more than 3 to 5% of the animal's body weight. This can limit what kind of technology can be used. So a lot of the satellite transmitters, which are really awesome and they provide real-time data, they are much heavier than what we can put on the kingfishers. So we used these data loggers, which are sorry, I just want, oops, go back. Can I get this to go up? Sorry, it's blocking that, but there were 1.6 grams, these satellite transmitters, and they were donated by the Swiss Ornithological Institute in Sempak, Vogelwarte. And these ones are really interesting because they don't just take light data, they also measure atmospheric pressure. They measure activity of the bird. Is the bird flapping or not flapping? They take a magnetism reading, and then they also take light. So they're called paneler loggers, or yeah. So over the course of 3 years, we were able to sorry. Forgive me. So we deployed 28 geolocators over the course of 3 years. Now, with these, you have to trap the bird again. You have to trap that same individual again after the bird has migrated north and come back again. So we were able to retrieve 5. But we were also able to confirm that 6 were lost. This is in the situation, we trapped the bird again and the bird did not have the geolocator on the bird. But because the bird is ringed, we know for sure that this is the individual that we put a geolocator on. So at some point in time, the bird must have lost the geolocator. The geolocator was placed on the bird, well, using what we like to call, it's kind of like wearing a backpack around the legs of the bird. This is different from the true backpack, which goes around the wings of the bird. But this was the method that was least intrusive to the bird. And this is what it looks like. So this individual is actually carrying the geolocator. This was after the bird was, this was days after the bird was released. We got an opportunity to see an individual and got a photo of it. Now, because they're cavity nesters, they go into holes. It is possible that maybe they just found a way to just get it and sort of squeeze it off of their back. We're actually not sure how they got rid. For those that got rid of their geolocators, we're not sure how they got rid of them. But we were able to retrieve 5, like I said, and this is once the data is downloaded, we were able to retrieve data from all 5 of them successfully. And in this particular case, this particular logger started recording in February. So think about each of these as a each of these as a vertical line, and that is 1 day, where the blue marks sunrise and the red marks sunset, and the dark part is, well, basically nighttime. So all of this data is sort of put together, and knowing what the sunrise and sunset is across latitudes, it is possible to then sort of determine where the bird was. But in the case of that's just in the case of a basic light detector logger. But like I said, these ones that were donated by the Swiss Ornithological Institute by Vogelwarte incorporated a few more nifty gizmos where we could tell activity data, was the bird still flapping or not. We could also get pressure data, which would give us an indication of altitude. The preliminary results, this I'm just showing for the results for a pair. So this was a male. And while this looks a little sort of squiggly, we were able to put that together to show sort of stopover sites as well. This is just an estimated route. The bird actually did not go this high, but the data just kind of we needed, this is preliminary, so after we cleaned it up, the movement of the bird was sort of capped close to where that 5 is indicated. And the larger the circle, the longer the bird stayed there. So this male carried the geolocator from January right through till December, when we trapped the bird again and retrieved the geolocator. We also have data from the birds, the female, which was actually the mate, the pair to this one. It had a slightly similar, a similar movement pattern, but different routes. They stopped at different places. And this bird also carried the geolocator from January 2017 to December 2017. It gave us some really interesting insight where my breeding pairs will not necessarily migrate together. One will actually leave before the other. Woodland kingfishers are solitary, and they're largely nocturnal migrants. So we're currently working on this particular paper now with the results on the 5 geolocators. And the sneak preview or the teaser is that we can actually say a little bit more now about how high they fly when they are migrating, their migratory flight. We can talk about how fast they actually migrate. And there's a bit more of information, because one of the individuals actually carried 2 geolocators. What I mean is, there was an individual that carried a geolocator, came back, we retrieved the geolocator, put another geolocator, and it carried it and came back again. And the bird's route was just, it was actually, it kind of used the same route. It was quite similar. So we're quite happy about that. I should also say that this part of the work builds on work that was done by Warwick Tarberton and Michelle. This was published in African BirdLife. And they put geolocators as on several kingfishers, and they got good data from a pair that was nesting in their home. And these birds went as far north as indicated, and the male and the female went to different non-breeding sites and came back down from there. But we're hoping to add to the data that they have based on all the other bits and bobs added to this geolocator that we were able to use. I'm keeping an eye on the time, so I'll just sort of try to speed through the rest. In terms of connectivity, we also looked at stable isotopes. What we wanted to determine was where the birds were molting. And what we figured out is that the birds in South Africa, the Cyanoluca, don't actually molt in South Africa. They come in and they breed, but then they go up north and they molt in what is supposedly their non-breeding ground, and then they come back south. So we had a master student take a look at this, and she looked at stable isotopes, deuterium, carbon, and nitrogen in the flight feathers of the bird. So she looked at the primaries, the secondaries, and the tail feathers. And what she found was that the isotopic signatures of the South African breeding birds, and we took the wing, the feather samples from them in South Africa. It was quite similar to those of the Ugandan birds, but quite different from the West African birds, the Ghanaian birds. So this does suggest that it supports that the Cyanoluca, at least the ones coming to breed in South Africa, are molting somewhere in the central Eastern Africa region. So this work has been, the dissertation is been submitted, and we're hoping that this will be published as well in some journal in time to come. So to just sort of close, I just thought to just show a little video, two videos actually. So this is the nest boxes that we used, but I should also say that the kingfishers used the standard box-like nest boxes. We also got some of them using those, and we got videos, and they were able to raise their young successfully.
I don't know, is it going to try to force the other one out?
In this particular video, there were actually one kingfisher was sitting and incubating, and the pair came in and kind of forced it out of the nest. And we were also able to get a small camera inside. So the bird was just coming in to incubate. And in this case, you can actually see the geolocator on the back of the bird. If you look to the back of this, so that's that little thing on the back of the bird. What we would like to do in terms of next steps is, as might have been mentioned at the beginning, I am currently based in Whitehorse, Yukon, but I would like for this work to continue. I would like to be involved in the future of this work, looking at intra-African migrants generally. So what we're trying to do now is focus on publishing as much as possible of the data that we do have. And part of this is, if there are students that are happy to look at the data and help make sense and publish it, we would be happy to sort of work with people and students in that regard. I know my strengths as a person. I love project design, I love field work, and I love proposal writing. Sometimes I get a bit distracted, or I'm a little bit slow when it comes to writing manuscripts. So I welcome all the help that I can get. But we also want to raise public awareness about intra-African migration and the need for such studies, we would like to fundraise. I'm sort of already looking at developing a number of proposals for work on the woodland kingfisher as a flagship species for intra-African migrants. But we would also like to expand this work. We would like to expand the network of sites where we're working. We would like to expand the number of species that we're working on. I should mention that we've also got some papers that there's a paper on the African pygmy kingfisher, because we also have data on that bird. We've also done work on the Diedrich cuckoo. So we would like to expand the work across species, and we would also like to expand the research questions that we're looking at. So if anything, the take home is that there is so much lovely, beautiful research to be done on intra-African migrants. And yeah, we're just scratching the surface, and we're already finding so many interesting things. Like I said at the beginning, it's taken an army of people to help get us to where we are. And I just thought I'd acknowledge some of them. This is not in any way all of them. A lot of people who have helped in the lab and who have helped be spending time in the given man hours in the field. And also to acknowledge many of our donors who have gotten us this far. We still feel that there is a lot that can be done and that can or should be done. So if anyone has any thoughts, any ideas, I am happy to discuss them. My email address is at the bottom. And with that, I would like to say thank you very much for your time, and I will welcome any question. Thank you.
Thanks so much, Dayo. That was a really interesting presentation, and you definitely have highlighted the fact that this is an interesting area of research, and there's still so much to do. I have a question before I start seeing what's in the chat. Have you thought of making use of the tour guide community that is pretty strong in many of those African countries that you're working in, like Uganda, for example, as some pretty amazing activity going on right now with tour guides, and they're out there all the time seeing things, and I think a couple of them are actually here this evening as well. And Kenya has also got it's got a good tour guiding community. Probably Zambia with Birdwatch Zambia. There's a lot of these community groups and so on that could help with gathering some of these data.
The short answer is yes. And we, even in selecting our sites, we worked, Malcolm Wilson, for example, to say thanks to him and to call him out. He was very helpful in providing advice on what sites, where he has seen woodland kingfishers. And we're happy to point this out to people, and not just the tour community in terms of the tour guides, but even tourists themselves.
Yes.
So we've talked about reporting rates in the Sabbath2 data, but even people who use eBird or whatever other platform you use, that kind of data would be great. And I mentioned Morphic earlier. Even if people are just taking photos and posting them, we're hoping that when we get around to getting the woodland kingfisher, for example, on the Morphic project, the more photos are out there, the more data we will have. And also getting to talk to people more, we can now make people aware of the differences, for example, between the Cyanoluca and the senegalensis and Fusiopelius. So when a South African guide is out and about, they can say, hey, look at that eye stripe. Do you know that, if they were leading maybe a group of people from West Africa, they could say to them that these kingfishers are different from your kingfishers, or something along those lines. But yes.
Cool.
And we welcome input from other tour guides. We're happy to work with people.
So there's a question in the chat, and if people have any more questions, please just type them in the chat. When you use call playback, do you use locally recorded calls to minimize contamination from non-local birds?
Hillary, thank you so much. That is an awesome question. So I will be honest and say that at the start of the project, we used sort of generic callback. But yes, after we figured it out quite quickly that we should use local. So what we would often do is take recordings from last season and use it sort of this season. And just to mention, sometimes what we also tried to do was have a particularly short call and a particularly long call. We call the short one the Pee Wee Herman call, and then the long call the Arnold Schwarzenegger call. And then we played with that because sometimes when we played a really strong call, some individuals will just not. It's like Arnold Schwarzenegger walks in. I'm not going to challenge him. I'm just going to walk. I'm just going to let him have whatever he wants. But in some cases, if you think you can actually take the challenge, then you will actually, step up. So sometimes we use the Pee Wee Herman call, but we also found that sometimes with the Pee Wee Herman call, the birds would just not even bother to respond. They're like, you're not even worth my time. So we had to sort of strike a balance. It makes it so much fun.
Cool. Any other questions, folks? I think you're reading the chat, so I don't need to read it for you.
Okay, so we have a question from Allison here saying, wow, so interesting and wonderful research. Thank you, thank you. I lived in Ghana for 28 years from childhood, and sad to say I didn't notice these birds then. Well, so in Gauteng, sorry, let me just expand this so I can read it all. So what's actually happening in South Africa, where you are actually now, Alison, is that I would say some 50 years ago, according to the Roberts Guide, the woodland kingfisher, then it was called the Angolan kingfisher, was just in the southern, it was just in a northern, northeastern tip, but their range in South Africa is expanding. And I've seen breeding woodland kingfishers at the zoo in Pretoria. So their range has come that far south. And this is largely because the bushveld habitat is largely being changed into woodland habitats, which is creating good nesting habitats for them. Now, this is good for the woodland kingfisher, but maybe we'll also have to consider, are we losing bushveld species as well? So that is not something I'm necessarily going to get into, but for where you are, you should be able to maybe start seeing woodland kingfishers soon in Gauteng. And from Colin, all the local guides I have reflect the woodland kingfisher as Halcyon senegalensis. Oh no, so I need to I'll make a quick correction, Colin. So these are subspecies. So the woodland kingfisher species, the generic name is Halcyon. The species or specific name is senegalensis, and then we have 3 subspecies. So it is, for example, the birds found in South Africa are Halcyon senegalensis cyanoluca. And then the ones in like Uganda, Halcyon senegalensis senegalensis. So we're going to the subspecies level to sort of split that up. So the bird guides are correct, but they are working at the species level, not at the subspecies level.
Any other questions? If not, I guess thanks everybody for attending, and hopefully we will see you in next month's webinar, which will be posted up on the site hopefully this weekend. And thanks very much, Dayo. Really interesting presentation, and I hope we're going to hear about the Diederik cuckoo sometime as well, because you may know that it's one of my favorite birds.
Yes. We're working on the Diederik cuckoo, not quite in as much detail as the woodland kingfisher because we couldn't put geolocators on them quite yet. But yes, we do have some interesting stories to share about the Diederik cuckoo as well.
Cool. All right. Thanks very much. And thanks everybody for attending, and we'll see you next time. enjoy the rest of your day, everyone.
Thanks, everyone.